This is working, or is it not? Is it working? Okay, so I hope it is working.
Okay. Right now we're going to hear from Liat from TEVA pharmaceutical company. It says, why is it important to know the mode of action of drugs?
So this is actually, I do want to say, firstly, well, it's my role today to kind of wake you up. So good morning, afternoon, or whatever. Thank you for inviting me to give this talk. I'm just, you know, it's kind of, how do you actually move from one slide to another? And do you have a pointer someplace?
No pointer?
No pointer. Okay. Technology, are we actually? So it's not actually just, why is this important to actually know the mode of actions of drugs? And we're not just going to talk to you about drugs today, but when you're going to actually apply to a very big company, to actually look at your files, to maybe sponsor your research or your idea, I'm going to be the one that will sit on the other side of the table and actually listen to you and know exactly and evaluate what exactly are you proposing. So I'm going to come from the other side of the table. I don't sound hungry because I'm working for TEVA for 14 years. And as somebody that is actually coming from in a very, very, very big company, we are always on the other side of listening to you, being the bitch that is asking you questions that most of the time you don't know the answer. Okay? So let me tip you some things for the roads. What is it important to us to know? Okay? From the other side of the table. So I just call it the mode of actions of drugs. But this is not just this. And we are going to talk about what is the importance of science when you're actually going to commercialize whatever you want to commercialize. It's very obvious that somebody was asking me to come from TEVA, that I'm going to talk about drugs, because we develop drugs in TEVA. We don't even, I have to say, TEVA doesn't develop any of the medical devices as well, only drugs, meaning compounds, active compounds. So I'm going to talk about drugs, active compounds, but definitely and certainly you can actually take it to any other subject or matter in the world that you, hopefully, the next inventors that will come with it. So this is actually the outline of my talk. I'm going to talk about licensing. What is important for licensing? What is potential safety, potential efficacy, and potential for other indication? And it's actually, I'm going to bring you some examples from some medicines that we developed in TEVA. What did we know? What did we not know? And why are we so careful when we are actually licensing you coming to us? Why are we so careful? And just explain you, why are we asking these questions? If you will actually understand the process of how we think as a big pharma, when you want to come to us for licensing, you will actually know to do the process better. So it's all about science. It's all about data. That's actually it, I think. So what is important for licensing? So when we are actually doing some licensing to new compound, what is it all about? So firstly, more we know, better we can evaluate its potential to be effective in patients. Okay? So if you know, if you have a compound, if you have a device, if you have anything, more you know about it, more we can actually evaluate everything else that has to do with it. The patentability is only one angle of it. Okay? Some of the things, if it's a molecule, there are some other like molecules that have been developed. Some else, some other people. We can evaluate it better, more data that we have. It is very important to for us to know if it has the potential to be unique in kind or a me too. What does it really mean? If you have a compound that you think that it will ever actually be active in a certain patient population, what is it actually doing? Why? Because if there are some other molecules that are maybe doing some other things, your molecule has a better efficacy, better pharmacokinetic, better brain distribution if you're targeting central nervous system, some other things. But if some other molecules that are alike are doing the same things, that gives us a lot of confidence. It's important for companies in order to build their pipeline, and people that are actually responsible for pipeline, for example, for TEVA, they can actually know that if it's a me too, for example, this is less risky from us. So if we're kind of building the overall pipeline, and if you go to any place to talk about, search what their pipeline is, because this is actually going to put you, if you know better about them, you know exactly what data you can actually give them. If they're only holding innovative drugs with a very, very high risk, they might want to listen to you. If you have something which is a new chemical entity, patentability is quite, you hold the patent for it, and the risk is maybe lower. That will actually give them a very good place in the pipeline. And the potential for other indications. Guys, if you know what the molecule is doing, if you know what the compound is doing, think about it always. Search very, very good. It might be much better than you think it is. So let me just give you an example. I just put here today, only because I gave another, I have to say, presentation at the other side of the university an hour ago. All the drugs that are actually marketed for Parkinson's disease. Interestingly enough, you need to be very high technology here to actually do here. But interestingly enough, look about this. This is a dopamine receptor agonist. It doesn't really matter for you to understand what dopamine is now, okay? It's really not. I just wanted you to point out these are marketed drugs that are already in the market. All of them are selling. That means that all of them are making money. This is a dopamine receptor agonist. This is a dopamine receptor agonist. This is a dopamine D2 receptor agonist. This is a dopamine receptor agonist. Look how many dopamine receptor agonists do they have here. All marketed, all making good money. Look at here. A lot of others. These are all in the market, and it's probably there is a place for all of them. What was actually surprising even for me when I actually made the list, okay, is that there are many dopamine receptor agonists under development. What does it actually mean? Guys, if you find something, and maybe some of them are actually with the same mechanism, that are actually doing some like things, that doesn't mean that there's not a place for your invention. Might be. All you actually see is where is your added value over the current existing, and actually offer it. Don't ever give up if you see that there are others. You see, there are lots of dopamine agonists in the market, selling already. Some of them are even generic and cheap, and a lot of companies are still developing them. Okay? What about potential safety? So again, it's a lot about science. So let me just, Eran was talking a lot today about kind of how do you, giving you some tips, how do you actually put everything together in the same place. And we always have to know that at least the practice of medicine is kind of an art. It's kind of a combination of many factors based on science. Okay, so data, information, mode of action is very, very important. It has not reached, perhaps never will, the dignity of a complement science with exact laws like astronomy or engineering. When we are actually talking about side effects, guys, sometimes a drug, as you can see here, it relieves watery eyes, runny nose, itching head, and scratchy nose. And the side effects include runny eyes, watery nose, the same thing. Because side effects in a lot of places, in a lot of cases, are only extended of their mode of action. So if actually something is doing something, okay, if it's doing it too much, it becomes side effects. So just take it into consideration when you're actually developing something. And it's all about kind of an overall concept, okay? It's always a balance between safety, efficacy, druggability, a lot of other issues that are actually there. The safety profile of a drug is kind of a composition of many, many things. More that you know about it, more that you know to look for it. And let me give you some tips in this so that you're going to be able actually to look for the right data at the right places. The mode of action of the drug actually tell us a lot about the safety of the potential safety or the potential safety problems of the drug. Where should you actually look for the information and come prepared? You have to understand actually, if you're actually doing something in a field, you have to actually try to actually get as much data as you can of it and never actually lose any piece of information. So if I take multiple sclerosis, MS is multiple sclerosis. It's a neurodegenerative diseases which have a lot of neuroinflammation. TEVA is developing few drugs for multiple sclerosis. It's kind of our core business. When you go to look for somebody that is going to invest money in your, look for somebody that what you are inventing is his core business. He will never actually, he will think twice before he's going to tell you to go out with your invention if it's his core business. He will always be afraid that somebody else is going to get your invention and it is his core business. So take it into consideration when you're actually going there. And always, always look for. Let me just explain you something like very, very easy. Some of the drugs in the market actually for multiple sclerosis are either DNA binding synthesis or some sort of or a modification of chemotherapy. Why? Because we have a problem here with autoreactive T cells. These are lymphocytes. There are too many of them. They are too activated, and I want to block them. Should I later on be very surprised to see that my patients are losing hair? No. Go for the mechanism. Look what the mechanism is. If the mechanism is identical to others, don't be surprised. And know that the person that is actually listening to you, he will not be surprised. He knows. Take, for example, there's a drug, fingolimod, is actually selling by Novartis. This drug is doing something very interesting. It takes all the lymphocytes in the body and actually put them in the lymph node in a way that they cannot get out. We call it in immunology putting your lymphocyte in prison. What's going to happen if you actually take all your lymphocytes and put them in a certain place and not available? What would you expect? Give me... I'm old. I cannot hear. If you don't talk loud, I don't have a chance. Go on. Immune suppressive effect. We know what's the role of lymphocyte. If we take them out, we know exactly what's going to happen, right? Good. So by that, actually, we know how to predict the adverse events. And later on in a clinical trial, we know how we actually observe them. There are going to be much more of them when the drug is actually in the market. The person that is sitting behind your desk that needs to give you money, he knows that. He already did all the range. And he knows that even if you're going to tell him, listen, but I took like 12 animals or 15 animals or 30 animals and nothing happened to them. And he's going to immediately ask you, how long did you treat the animals with? Did you expose them to any infectious? No, you did not. Expected with this mechanism of action, this is where actually you're running a bit short, I would say, with the data. So we have the predicted AE, and then we have the observed AE, which are not actually different. And listen to this. Well, of course that lymphopenia is one of them. But why is it not surprising? What did I know before? That I'm actually taking all the lymphocyte away. If this is what I do, this is actually the side effect, I actually should know that in advance. Let me give you some other, example. For example, another drug for multiple sclerosis. Multiple sclerosis is we do a lot of research in this. Is actually currently, I have to say, starting to make a lot of money in the US, and they actually just got approval in Europe. This is going to be blockbuster. Blockbuster is a drug that is making over $1 billion a year. Many, many years ago, it was published in Brain, not so many years ago, in 2011, that this is actually acting on a specific pathway in the brain. Doesn't really matter now. You don't have to understand the specific pathway. Just want you to know how can you actually look for similarities. At the same time, there's another drug that somebody else is actually developing for kidneys disorders. But same mechanism. And then look what happened. So what happened is a clinical trial. The clinical trial with this drug with Abbott that get this mechanism to the kidney. So they had 60 patients die in a very, very short time. And the FDA immediately halt their system and everything. Immediately, they stopped the dosing immediately. What else happened? At the same time, almost simultaneously, the FDA paused for a brief their assessment of BG12 as well. Why? Because the FDA knows that if you act on the same system, and here they have 60 patients death, what's going to be here as well? So if you have something that is actually active on a certain system, look and dig very carefully. What happened in the past? Every patient exposed to it? Do we know anything about this? If you don't know, somebody else will. What about potential efficacy? And if you go actually and look and you have something that you think will be active at something, okay? Why is it very helpful if you know exactly what does it do? And not just estimate, that you actually know what's the mechanism of this compound, vehicle, whatever you're actually trying to invent. So firstly, we have to know which elements in the disease pathology does the compound target, if we're talking about compound, for example. If you are looking for something that should be active, for example, in Parkinson's patients, Alzheimer's, multiple sclerosis, all, I would say, brain disease, could you please check before that this compound has the ability to cross the blood-brain barrier and get to the brain? Will be helpful. Sometimes you come to us, and then, you look at this and you say, but I have something, it's so active, I give it to cells in vitro and it works great. Needs one look to say, yes, but it's too big molecule. It will never cross the blood-brain barrier to get to where you think it should be active. So bear in mind, all I'm saying, bear in mind. What should be the primary endpoint of the clinical trials? What is your compound doing? If you're not going to tell me what is your compound doing, how should I, think about designing clinical trials, identify patient population? Give me the clue. You're the scientist, or at least you want to take the science or whatever and translate it into human use. And can we use it in combination with other therapies? Why is this important? Because if you don't know what the drug is doing, the compound, the vehicle, whatever you are trying to, if we put it sometimes with other, you have to think, I don't know how many of you know very kind of old people. So for you, I am a very old people. But, you know, not actually for me, but when we're actually considering older people, we are considering people over 65 or 70. Do you know how many drugs do they take a day? Plenty of. So you have to actually see, I mean, if you don't know what the compound is actually doing, you will not be able ever to think about combinations. Let me take you through a story, because a story is always good to tell. This is a small molecule. TEVA actually, anybody has questions? You sit very, I mean, do you sit very silently because I'm boring you or because you don't know what to say? What? I did listen very carefully. Okay. That's okay. If you want me to talk, I mean, if you want to ask questions or anything else, just, you know, because you're really too quiet. Never actually, I was just actually giving a talk on the other building. So there were a lot of professors, and they were talking actually all the time. So it's they almost actually didn't listen, I have to say. So that's a small molecule. TEVA bought this molecule from a Swedish company with the name of Active Biotech. We got into licensing agreement with them on this. To tell you that I think that it was the best, I would say, deal of the year, but they offered it for multiple sclerosis. And multiple sclerosis is something that we do. And there's always kind of a threat, okay? It's our expertise. We wanted to have a big pipeline, and you don't want other people to buy it. Maybe, you know, even if it doesn't worth so much, it's better off that you're going to have it than somebody else, okay? So take it into consideration. We bought it. We thought that it's a novel, once daily. Oral at that time used to be a very good advantage, not anymore. When you look and you want to sell somebody, try to see who else is developing what. Because at that time when we bought it, I think it was 2007, I have to say I don't remember anymore. All I remember was that I was in Sweden, and then I was in Switzerland in an airport, and we were actually reporting on the due diligence, and then the decision was made, and by the time that I flew from Zurich airport to Tel Aviv, it was announced already in the news, and we kind of missed it in a 4-hour flight. But to tell you, I mean, I travel so much all the time, I don't know anymore, remember where I was, when, why, etc. We knew at that time that it's an immune modulating agent. What is an immune modulating agent? It takes the immune system and it modulates it. This is actually what was published at the time we bought Active Biotech drug, laquinimod, for multiple sclerosis. All they actually, you know, gave us is, what's the mode of action? What is this drug doing? It shifts the immune system in the periphery. Good. We know and understand, and we investigate the research in multiple sclerosis for many years. Because we have Copaxone for over 20 years, the research of multiple sclerosis is something that we are very much aware of. And we know how does the roadmap look like. When we actually investigate multiple sclerosis or Alzheimer's disease or Parkinson, whatever, you know, everyone has this yellow brick road. And we are on the way to the Wizard of Oz, okay? Always think about this and know exactly to identify your yellow brick road. Why? Because this should be your Bible. This is how it looks like for multiple sclerosis. Every one of you is going to do it for a different indication, for a different disease, for a different thing. Every one of you that is actually dealing with something, try to see the overall picture of the patients and overall pathologies if you're actually looking for a specific effect in a specific disease. Now we know multiple sclerosis. We know that multiple sclerosis consists of two major elements. These patients are relapsing, remitting all the time, and wherever they actually do a relapse, they actually hospitalize, and it's very bad. But at the same time, there's kind of an ongoing neurodegeneration that at the end will bring these patients to a wheelchair. What is actually responsible for what? It's a big question, I have to say. And via actually the research on laquinimod, we actually discovered a lot of other things. So, knowing that laquinimod, by the time that we had it, this is how we bought it from Active Biotech. This is what we got as the main mechanism of action. The main mechanism of action that it was offered to us was actually here in the periphery. We know that if you have drugs that are affecting the periphery, you are affecting the relapse rate in these patients. So we designed a very big 2 clinical trials. We had a phase 2 clinical trial. This phase 2 actually showed us some effect on the relapse rate. But phase 2, as you probably know, is only part of it. It's kind of a small, and it's more exploratory. And then you go to big, 2 phase 3 clinical trials. Do you know how much money is this slide? There are 2,000 patients here, 156 centers around the world. Do you know how much money it is? Go on. Almost. This is actually the disposition of the patients, which is not important to you, but what is important to you is the primary endpoint of the clinical trial. What would we choose as a primary endpoint? The relapse rate. If the compound is actually affecting the lymphocytes, then it should actually affect the relapse rate. And I will show you later why did we actually choose it and how is actually the MS market look like. What other drugs are actually we are experienced with. We had all the rationale in the world to actually choose this as a primary endpoint for the patients. At the same time, we started investigating the mechanism of action of the drugs. When we are actually doing this, we are really doing a good work. We are industry. We're not actually playing, and we don't have time, but we do have money. So we actually put a lot of effort of understanding every compound that is actually under development very thoroughly. So we actually went through all the development, looking for one factor after the other to see where is this compound effective. Why? Because of all the reasons that I actually showed you before. So we went actually to everything in the periphery. We knew that the permeability of the blood-brain barrier is very important. It's important that the blood-brain barrier is going to have the ability to actually get your molecule from where you take it, from the periphery, and transfer it to the central nervous system. And actually, we looked at the pathology in animals of what is this drug doing in the central nervous system. We do research all over the world. You have to always know that the people that are actually sitting in front of you and the other side of the table, they are actually serious, as Iran just said before. They know what they do, even though we don't look this way. And this is actually what we found out. It was a surprise for us, I have to say. So this cell is a very, I would say, different cell. It's a resident cell in the parenchyma in the central nervous system. This cell is actually composed most of your parenchyma and most of your area in the blood, in the central nervous system in the brain. It has very strange characteristics, I have to say. It's, and definitely I can tell you, so I published this paper in 2012. And I got an editorial in the volume emphasizing the importance of this cell in this disease. So it was not so much known before. This is a kind of a very simple thing. I'm just going to get you through. These cells are actually part of, really consist of over 90% of your parenchyma in the central nervous system. I call them the beating husband cells. Why? Because when they are angry, they destroy everything around you. But then, when they're actually calmed down, they bring you flowers. So what they're actually doing is once they are being activated, they are really very destructive. They're actually destroying all the neurons, the axons around them in the central nervous system. So there's a very massive tissue destruction following the activation of these cells. Who activates these cells? Inflammation. It's actually kind of a commonality pathway. Later on I'm going to talk about commonality pathway. So it's a very interesting cell. Later on it secretes a lot of beneficial, I would say, after the damage. It secretes a lot of beneficial factors that actually give a lot of nutrition to the neuronal whoever left, I have to say. And what I actually found out, I have to say and admit, that it was completely serendipity. We found out that the main activity of our drugs is actually lying down here. So we actually started on possible activity in the periphery, found out that the most affected cell is actually lying down in the parenchyma, in the central nervous system. So what is actually the compound is doing is preventing these cells to get this star-shaped cells, but they get lean and mean, okay? And they actually secrete factors that are actually destroying all the surrounding area. If you prevent them to get in this state, you're reducing significantly all the damage to the surrounding area. This is the NF-kappa B pathway. I'm not going to get you through here. It's a very high science, and I have to say probably not all of you are medical and biologists. I'm just going to actually skip it a bit, just to actually tell you that we identified the path very, very thoroughly, and it was a very hard piece of science. I'm actually moving forward to getting the... Okay. So we found out about this drug, that it's not actually protecting the neurons from damage directly. But what it does is that it prevents these cells that are actually, when they are getting activated, they're actually destroying and making a lot of damage to the surrounding area. So that's going back to the map of multiple sclerosis. This is, as I told you, that's for the people that are working for multiple sclerosis, and whatever you're going to do in your life, you have to have a map. You have to know all the factors that are actually consistent in what exactly are you working in. And we actually found out, just to remind you, we bought this compound, we started here. And we ended up thinking that this is actually the most significant target. Why is this a problem? Okay? Now that you actually know that I would say, affecting the periphery is affecting the relapse rate directly. And now you actually say, this is not it is a target, and laquinimod is active there, but it's much more active someplace else. What is it actually affecting immediately? How do you design your clinical trials? What's your primary endpoint always in a clinical trial? Your primary endpoint is where you think your compound is going to be the most effective. So did we choose the right primary endpoint to our clinical trial? Would you do different? Is it yes? Is it no? Is it I don't know? What do you think? Do you think that now, when you actually know that the mode of action, or the main mode of action, lies someplace else, what would you change in your clinical thinking and your clinical design?
To
Okay. Anybody else? So we actually learned that this drug is actually effective mostly in the central nervous system. And if we are actually, at the same time, by the way, we opened the clinical trials and we saw that we do have an effect on the relapses, but it was much less significant than the effect that we had in the central nervous system, preserving the tissue loss for the patients. So we actually went again. I'm not going to get you through here. It's just to actually explain you how do you actually treat your competitors, what else you can learn from looking very thoroughly on the market. And this is kind of the current map of multiple sclerosis. So I'm not going to get you there, but I will just get you Okay. So we actually, when you look at the MRI of the MS brain, okay, so this is actually where, if you are affecting the periphery, this is where you target. The pathology in the brain is consists of two very major pathologies. One of them is the relapse-related, and the other one, which is much more significant because this is actually less than 1%. The other one is more than 95%. It actually can be influenced with the compound that is getting into the blood-brain barrier, crossing and getting into the central nervous system and affecting local inflammation in the brain. So we actually thought that this is actually the main thing. We came out that this is actually the main thing. So what we went again, designed another clinical trial, another phase 3, when we actually switched the endpoints from secondary to primary. Won't tell you how much money did it cost. Potential for other indications. I still have like another 10 minutes. Okay, so we know. Nice. Bit too late, I would say. Could do better. Then what else do? You actually said choose your battles. Now, if you know an effect, go for other indications that might actually benefit from the effect that you know that you have, right? This I would actually do only if I would really, really have a very good and solid data. So be careful actually what you're wishing for. But once actually we know that this is where the main mechanism, where it actually lies down. That was easy. Cost us a call to somebody that is a neuropathologist working in Germany, and they have a brain bank. Brain bank is not something that is very nice, for you to think, but you should actually know that things like this exist. Why? You have to know where can you get information and who actually can help you or think for you, as Iran just said. And what I was actually asking him to do, can you actually get me 4 neurodegenerative patients in 4 different diseases and see what's the role of this specific cell in all the 4 of them? And we came out with this. And he actually was surprised as well. He said, I never knew that this cell is so affecting in all these 4 neurodegenerative diseases. He couldn't make the difference. So when you're actually talking about other indications, and try actually, when you sell your compound invention or whatever, try to think about this yourself. Otherwise, the people that are actually buying this for you, they will think about this, and they will actually develop it for a lot of other indications, and it's very important for patent as well. So we actually looked a bit, and we found out that Huntington disease has the same performance of these cells, and not just Huntington disease. This is actually a very early event in Huntington patients. Alzheimer's disease as well. It's an early event in Alzheimer's disease as well. ALS disease. And a lot of other indications that has to do with neurodegeneration. So from knowing the mechanism and from knowing what is your invention can do, you can actually end up with a lot of indications, with a lot of opportunities with actually what you can definitely do. With this, I will finish. Thank you very much for listening to me. And if you have any questions, please let me know.